Good afternoon, everybody. I think we'll get started. We might have one or two people drift in, but we have a lot to talk about today, so let's get our talk underway. My name is Kara Fligg, and I have the pleasure of working at the McGovern Institute for Brain Research here in this building. We are especially pleased to welcome folks from the class of '59 today. That was Pat McGovern's class, and many of you probably knew Pat, together with Lori Hart McGovern, his wife, who's right here. They founded the McGovern Institute for Brain Research in the year 2000. We now have 19 faculty members and more than 300 scientists and engineers from many departments across MIT, all devoted to understanding the brain. McGovern research ranges from studying molecular genetics that play a role in brain disorders to understanding the neural circuits that underlie learning and behavior, all the way through to studying the complexities and nuances of the whole human mind. We also engineer breakthrough technologies that are used by thousands of scientists and clinicians all around the globe. I'm very pleased today to introduce our director, Bob Desimone. Bob joined MIT in 2004 and is, in addition to being director of the McGovern Institute, he is also the Doris and Don Berkey Professor of Neuroscience in the Department of Brain and Cognitive Sciences. Prior to joining MIT, Bob was director of the Intramural Research Program at the National Institute of Mental Health. His own lab here at McGovern studies the mechanisms of attention. Today, Bob is pleased to share with you some of the strides being made at McGovern to understand and develop new solutions for complex brain disorders. Please help me welcome Bob Desimone.
Maybe we should take some of the signs off the chairs and let other people just come in and fill in. Well, welcome. It is great to have you here. It's a really special couple days. I'm going to talk about what we know about brain disorders. I'm going to use some of the work from the McGovern Institute to highlight these stories. But you know, you can regard this like an MIT classroom. You know, it's better if you interrupt. If it'd be a little interactive. No, really? You want it all like digested and fed to you? Just passive recipients? Okay, all right. That's what it was like when you were here. Not anymore. All right. Well, and we had reserved these first couple rows for class of '59, but they're not all full. So feel free, if you're in the back, to take a class of '59 honorary seat. So as Kara mentioned, the McGovern Institute was founded by Pat and Lori McGovern, and we have Lori McGovern here with us today. In fact, Lori is at Lori's at many of our events, and I think are you an honorary class of '59 member? I think they've accepted you into the fold. Oh, very good. And when it was founded, it had a dual mission, which was to first understand the brain. This is our basic research mission. It's always the foundation of what we do here. And then, but the second part was to take what we've learned about the brain and to try to apply that to help people suffering from any form of brain disorders. And also we try to help in education and development of technology, and some of those are different stories. The approach that we take here is a multi-level approach. So we go from genes to cells to circuits to cognitive function and even computational neuroscience. And our faculty members, who are shown here, span this whole range, and you'll notice that many of them actually are sitting at the intersection of these different levels because their work bridges across the different levels. And we have a very distinguished faculty. We have the ones highlighted in blue are members of the National Academy of Science. We have one Nobel Prize winner, Bob Horvitz. And I have to say it's really been a privilege for me to work with them since I've come here to MIT. Now the problem that we are turning our attention to is just a really, really serious problem in health, which is all the various forms of brain disorders which affect people. And of course, they not only affect the person with the disorder, but their whole family. You know, it's everyone's drawn in. And I would like to be able to tell you that, of course, we've been making steady progress, and there's fewer and fewer people diagnosed with brain disorders every year, but it's the exact opposite, right? There's actually more and more people diagnosed with various forms of brain disorders every year. And to a certain extent, it's just better assessment. We're getting better and better at recognizing things that affect people's brain, affects their behavior. And also, I think we are making some progress in reducing the stigma of brain disorders, so people are more willing to admit it, and so they come to our attention. But then once they come to our attention, it's just staggering how many people have some problem affecting their brain. Of course, you know, we think all of our behavior is coming from our brain, so it's not as though, you know, we have the other behavior that's not affected by our brain, right? Everything's affected by our brain, and we can identify various ways in which it may not be working optimally. If you look at the impact of brain disorders, which is shown here in this graph for the two, for both neurological disorders and mental disorders, as a proportion of all the different impacts on what they call disability-adjusted life years, which are all the days of disability that are affected by, and you name it. And the thing about many of the forms of brain disorders is they start when you're young and they impact you for your entire life. You may be dealing with some of these problems for 50 years. And whereas in some other diseases, unfortunately, people may die quickly, but it means that they're not suffering from a disability for 50 years. And you know, it may be far less. So the disability impact is huge. And you know, you just pick up the newspaper these days, or actually, I guess, pick up your Kindle these days, and read, you know, what's happening in the health field, the medical field, and of course, there's all the dramatic improvements that are being made in many areas of health. And you know, we all know that, you know, of course, the most famous examples, the incidence of polio, basically, you know, almost wiped out. You look at the incidence of HIV, more modern problem. Deaths from HIV dramatically falling. You look at deaths from cancer. Another, you know, it's not as dramatic as some of the changes we've seen in HIV, for example, but there's steady reductions in deaths from cancer, deaths per 100,000 population. And this graph stops early, but this decline has continued and is projected to continue off into the next few years. And so you could ask, so what's responsible for all these improvements that are being made in other areas of healthcare? Well, they're based largely on a better mechanistic understanding of disease. And you know, I'll just get, you know, just one of many examples is the chronic myelogenous leukemia, which was identified actually many years ago as being associated with a genetic mutation in a tumor. And having identified the genetic mutation, physician scientists were able to identify an altered gene product. And then having that gene product, they were able to develop a drug, Gleevec, one of the great success stories of modern medicine, that could block the action that was causing the cancer. And has essentially eliminated this disease. And so it is especially frustrating that we're not seeing the same kind of progress in brain disorders. And you ask, well, why? So why is it everybody else gets the benefits of everything we're doing in medical research, and why aren't we getting it in brain disorders? And the answer is we do not have that mechanistic understanding. We do not have the identified gene product, the target for the drug. If I asked anybody now, you know, what's the mechanistic cause of schizophrenia? We don't know. What's the mechanistic cause of depression? We don't know. People think that we know because we have some drugs that work through certain neurotransmitter systems, but there's absolutely no reason to believe that those drugs work because the problem is in that neurotransmitter system, right? Aspirin works for, when aspirin reduces your fever, it's not getting at the cause of the fever. It's reducing a symptom of the fever. And that's the problem that we have in brain disorders, and that just means we're further behind in some other areas of medicine, and we have a more difficult problem, right? It's a lot easier to understand what's, say, going on with your liver or your kidney than it is with your brain. And, when a patient goes to their doctor and they have a kidney problem, it can be examined, and if you have a tumor, you can take pieces of it out, but it's not very often you're going to go into your doctor and he's going to take out a piece of your brain and examine it, right? And so it's a much more difficult problem. And so we're further behind, and so we need to gain that mechanistic understanding. And if you look at, what's the roadmap? How do we get there? Okay, and the roadmap really comes from all the other areas of medicine. You identify genetic vulnerabilities. You use all the new tools that are being developed to try to understand how you can go from a genetic vulnerability to an altered emotion or brain cells that are dying or whatever. We have a better understanding of how those genetic vulnerabilities lead to changes in our brain circuits. And we develop animal models for testing, developing and testing new treatments. We use tools like brain imaging in people, which is our non-invasive way of looking at brain activity. And then with all that in hand, we can then begin to develop new treatments. And so the roadmap's clear, but we'll talk a little bit about what's the reason for hoping that we are going to make progress following that map. Well, I told you, so that the starting point for a lot of this work today is our understanding of the genetic vulnerabilities that lead to a lot of these brain disorders. And the work that's going on in genetics has totally revolutionized this field in just the last few years. A lot of the work is being led by the Broad Institute across the street, and they're close collaborators of ours. And they're doing what we've learned about genetics and disease is that we used to think that you could be able to do genetics on a couple dozen people, find some big mutation, and then you end of story. You've got your answer. But it turns out that the genetic effects from any individual genes are usually small, and so we need many thousands of people in these samples. We did not understand that just a few years ago. So there's a rush around the world to collect these very large samples of people. 100,000 people is not too many in some of these genetic studies. And they're paying off. They are identifying genes that contribute to these vulnerabilities. These are just some of the confirmed psychiatric risk variants, up until a few years ago. It's going way beyond that now. We now have identified hundreds of genetic vulnerabilities, and one of the really surprising things that's come out of this is that many genes that lead to vulnerabilities in one form of brain disorders is leading to vulnerabilities in other brain disorders as well. And so you might see, in a family, you might see depression, but you might also see autism, or you might see schizophrenia pop up across the generations. And we're beginning to understand that some of these things have shared vulnerabilities that we need to understand. These are just some of again, this is from a few years ago, but numbers of genes that have been identified that are associated with, in this case, psychiatric disorders. Okay, so what are some of the new tools we have available for understanding the impact of these genetic vulnerabilities on neural circuits and brain function? I'm going to give you a couple examples from the work of Ed Boyden and Feng Zhang, two of our faculty members here. Ed, I'm putting up an example from expansion microscopy because this is one of his latest discoveries. Ed has discovered a way to essentially blow up brain tissue so that things that were formerly very difficult to visualize with microscopes now become visible, even with conventional microscopes. And what you saw there, a piece of expanding brain tissue, he's figured out a way of using some of the materials that go into baby diapers. You ever have a baby diaper? The fundamental principle of a baby diaper is you put a little bit of liquid on it and it blows up, right? So he figured that if you put some of the same material into brain, it will allow the brain to blow up when you put it in saline, and he does it in a way where all the microstructure is preserved. I mean, down to the nanometer, the structure is preserved. And I'm just going to show you, this is an example. This is a neural circuit, and some of the little dots you see in this circuit are the synapses between the individual cells, which we're now able to visualize in really unprecedented detail. I think when this slide was made, I think Ed was up to 5 times linear expansion. I think he's now up to 20 times linear expansion. It's growing dramatically. And not only are we able to visualize the structure, but we're now able to tag these with genes that are active, for example. And we're able to do this in both in normal control populations, disease populations, and so on. This is totally revolutionizing our ability to localize where some of these changes are affecting function in brain. Another really revolutionary method that Ed co-developed is what's known as optogenetics. Now, in optogenetics, you insert some genetic material into neurons, and so this is why it's been done mostly in animals so far. But then having inserted that genetic material, neurons become sensitive to light. I think I can show you that video again. Oops. What's that? There was algae. So it's using material from the natural world that gave algae light sensitivity. And Ed and his collaborator Carl Dijsselhoff and Feng Zhang, when he was a graduate student at Stanford, decided they would test this in neurons and discovered that it worked in neurons. And now using light, usually laser light, you can now activate individual cells or different cells and populations of circuits, and for the first time really test the function of some of these neurons in these circuits and understand what's going wrong in genetically defined subtypes of neurons. And there's a lot of interest in using this clinically. Of course, the hesitation is for brain. We don't want to inject a lot of genetic material into brain. So currently it's mostly being used in eye disease. There's already clinical trials underway to restore blindness in people by making some of the non-receptor elements in your eye sensitive to light. And now you could bypass, for example, damaged photoreceptors like you have in retinitis pigmentosa. And this is under clinical development. And there's some other peripheral applications, but I think what we'll see over the next 5 years are some applications in brain as well. But because it's just an invasive method, Ed has also been interested in developing non-invasive methods that can be used in people for stimulating brain activity. And not just as a research tool to understand how the brain's working, but as a therapy. And many of you know about deep brain stimulation being used in Parkinson's disease, for example, where you have an electrode implanted and it can completely turn off the tremor that you see in someone with Parkinson's. And there's increasing interest in using the stimulation approach to push the brain in different directions that will be beneficial in a variety of forms of brain disorders. So what Ed started to do was to use electrical stimulation from outside the skull, but usually when other people have done electrical stimulation from outside the skull, the problem has been that it's not very focused, right? It just, you put a pad and you put electricity and it's going to go all over the place. And so what he's been developing is ways of focusing the stimulation using different frequencies from different electrodes. And then by calculating where the interference pattern should be at different frequencies, he can tune where the stimulation is affecting the brain. So for the first time, we have a way of targeting with external stimulation, targeting individual parts of the brain which can now be stimulated. And this is an active development as well. And this is now an MRI image of people that have had this stimulation applied, and these colored spots are the areas where the stimulation activated brain activity. You can see how it's much more focused than just this diffuse state that we used to have before. And I think this is something that will also have direct clinical applications. Now going way over to the molecular scale, I think everybody these days has heard about CRISPR. CRISPR methods for doing genome editing. Basically, the ability to edit the genome the way you would edit text with the word processor that was developed in cells by Feng Zheng here. And in fact, just today, I don't know how many of you saw the news reports, just today, a new paper came out by Feng, reported in, it was either Science or Nature, I think it was Science, reporting a completely new way of doing this genome editing, but in non-dividing cells. The original method that was developed required dividing cells, which would be useful in developing animals or possibly developing people. We won't go there right now. But your brain is full of non-dividing cells, right? So it's no longer dividing, and so it's been a real challenge of how you might use CRISPR on brain. But now fungus has developed a new way based on jumping genes for inserting new brain material. Yeah?
I've heard the phrase neuroplasticity, which sounds like the brain is actually capable of being more alive than we have thought. How has that insight been evolving?
Good question. So, we know our brain is plastic because you did get an education here at MIT, right? And I guarantee you it was changing your brain. And so that kind of plasticity, that's natural. We're all born with that kind of plasticity. The question now people have been asking is, how can we use some of these plastic mechanisms of the brain and hijack them for other purposes to get them to solve some of the problems the brain has using all new ways? So for example, in my own lab, one of the things that we're trying to do is to change how people pay attention and change their ability to pay attention using neurofeedback based on using what's known as magnetoencephalography, picking up brain signals at very high resolution and giving them that feedback and the ability to control their own attentional state. So we're hijacking the brain's natural plastic mechanisms, but by giving people a different kind of information about their own brains, which they never had before. No one could ever gain insight into what's happening in your own brain. But by giving that as feedback, actually enabling people to use these plastic mechanisms maybe to overcome a problem. So it is. We are trying to make use of the brain's neuroplasticity for solving some of these problems.
In the Venn diagram exhibit that you had showing the multiple disciplines, how much biochemistry is part of your thought process? And I was thinking that might be at the molecular or cell level, but could you
That's
say
right.
something about
Well,
that?
biochemistry is extremely important because, again, we're trying to understand the actions of genes. Genes work through other molecules inside of cells. Those are mostly biochemical pathways that are being influenced by these genes. And not only is it giving us some insight into how different mutations affect these pathways differently, but if you're going to develop a drug therapy, we'll get to that in a moment, if you're going to develop a drug therapy, you're generally looking at ways of getting molecules in and influencing these pathways and so on. What methods like CRISPR are doing are at least raising the possibility that in the future there will be non-drug therapies. There will be biological, but not using drugs in the way that you normally think of them. We'll be going in there and manipulating the genes directly. And some of you may have recently seen some of the new approaches in spinal muscular atrophy. It was a new gene therapy approach that was just approved. And there may be many more of these things. Right now they're not very many. They're incredibly expensive. But this is the very beginning days, right? This is all going to change in the next few years. I'll get to that in just a moment. But CRISPR gives us a research tool and potentially a therapy. And I think one of the reasons that this becomes so important in thinking about, say, treating a disorder like autism is we now understand that disorders like autism, they may have a common name, but they don't necessarily have a common basis. And so there could be different genetic forms of autism. And if you're going to go in and try to help someone with one genetic mutation, it necessarily isn't going to help somebody with a different genetic mutation. And so it's going to lead us really into the era of precision medicine for brain disorders, where it's not a one medicine treats all. It's going to be identifying what's wrong in that individual patient and targeting what's wrong in them. And autism is certainly an instance of that. One of the things that Fung has been developing that could be used in brain potentially now, and it's under active development, is instead of targeting DNA, which has been lends itself more easily to dividing cells, he's targeting RNA, which is the product of genes. And RNA you can target without cells having to be dividing. It can be done in adult cells. And I think that's where most of the action is going to be in all these brain disorders. We're not going after embryos, we're going after adults. Adults or children that are born, they have a problem, but we're still going to be able to go in and manipulate gene products to help them. And RNA editing is a possible way of doing gene therapy in adults. Another really, to me, very surprising application of CRISPR methods is actually in diagnosis and detection. And Fung has developed a new method called Sherlock, some of you may have heard about, for detecting incredibly small amounts of genetic material with very simple methods, almost like the pregnancy test. Very small amount of some, could be saliva, could be blood, could be urine, in which you detect tiny amounts of genetic material, very simple devices. And so it's being used right now, for example, for detecting Zika virus in populations where, in a lot of places where Zika is developing, they don't have access to very sophisticated genetic sequencing machines and so on. Very expensive, you need sophisticated labs and so on. But with using these CRISPR-based methods, they can have very inexpensive testing devices and very, very rapid detection of very tiny amounts of infectious material. And now it's also being applied to detecting genetic mutations in people. Initially, it's going to be in cancer. You have some very simple tests. We'll identify whether you have a mutation associated with different types of cancer, and then we're expecting this to be used in brain disorders in the future as well. So simple testing, precision medicine, identifying what's wrong in each individual person, and then targeting what's wrong in their treatment in that person. So now I've been telling you a lot about what's going on at the gene level, but of course genes themselves don't do anything except by influencing the neural circuitry in our brain, right? That's how we get behavior, that's how we get thinking, that's how we get thought. It's all based on neural circuits. And one of the people that is one of the world leaders in understanding the function of neural circuits is Anne Graybill right here in our institute, who has been studying neural circuitry in animal models, and I could give you many examples from Anne's lab, but I picked this one of the more recent ones where she studied the neural circuitry involved in the kind of decision that we are making all the time. Every day we're faced with decisions where you're balancing risk versus reward. So in this case, she was studying mice who were going for different amounts of sugar in cups, but some of them were associated with a bright light, which is very aversive to a mouse. They don't like being in bright light. And so they had to weigh, well, is it worth, that amount of risk of a bright light by going after what I think is actually the bigger reward down this alley? Or should I take the safe route, which is not associated with bright lights, but is associated with lesser reward? Always a balance. And what she's identified is a circuitry that seems to be adjusting between those two alternatives. And by manipulating the circuits in one way or another, she can nudge the mouse to take more risk in one circumstance, or nudge the mouse to be more cautious in another. And we think that, yeah.
This is the same mouse?
The same mouse. She can alter the circuit in two different ways, but depending on which neuron she's targeting, and get it to do one versus the other. And this is really, a lot of this decision-making that we face is really the essence of a lot of our anxiety, right? When you take the riskier alternative, you're more anxious. And it turns out that some of these circuitries are sort of naturally biased towards this generation of anxiety. They make even normal decisions seem risky. And it generates a lot of anxiety, and so it's like the circuitry needs to be sort of shifted back into a less risky analysis of that situation.
How does she adjust the circuitry on a living subject?
Well, in mice, she can do it with optogenetics. So just that method I told you with the shining the light, so she can insert these tiny fiber optics with the laser targeting genetically identified cells, and then by targeting with one color of light or with one fiber, she can influence one group of cells, and then with another fiber or different genetic targeting, another group of cells. It's really remarkable. It's like playing the piano, but with neural circuits. Another approach that Anne's taking in collaboration with one of our other scientists here at MIT, Bob Langer. Many of you have heard of Bob Langer. Anne's been working with Bob Langer for developing these microprobes for both monitoring neural activity and injecting tiny amounts of drugs in a specific circuit. And I think this is going to be another approach in the future. Right now, you go to your doctor, he gives you a medicine, you take a pill. It's affecting your whole body, right? And that's why, and so you get a medicine, and then you may have to take 6 other medicines to control the side effects of some of the other medicines, right? It's you're always chasing the side effects because nothing's targeted. There's all these off-target effects. So what Anne is trying to do is develop these probes with this collaboration with Bob Langer that would target the specific circuit. So she identifies the circuits in her studies in mice, and then the idea would be, if you understand the circuit problem, develop this kind of implantable probe that could deliver the medicine to just the one place where it needs to be applied, and not bathing the whole body in a chemical that may have untoward side effects. Now, another application of the CRISPR methods, I mentioned animal models, but is in developing better animal models, and particularly primate models. It's something that we've become very interested in. And Guoping Feng is leading this effort here, and we have a paper coming out next week in Nature reporting the first findings from developing these genetic models in primates for a specific form of autism. It turns out that these genetic studies that are identifying the vulnerability genes are discovering that a lot of the vulnerability genes for brain disorders are affecting proteins in the synapse. And the synapse is how the neurons communicate with each other. And it turns out that that communication channel is where a lot of the problems are coming from. And it's incredibly complicated. You think about just, a synapse is one thing up against another. It's not like just electrical coupling. many, many, many complicated proteins interacting in ways of bringing molecules into a cell, getting to nucleus, affecting cell machinery, and so on. And so there's basically a lot of things that can go wrong. And so a lot of genes can influence the synapses in bad ways. And one of the mutations that's known to be associated with a form of autism is in a gene called SHANK3. It leads to a disorder, a specific subtype of autism known as Phelan-McDermid syndrome. And the single mutation in the SHANK3 gene essentially gives you this disorder. It's like virtually 100% of the people with this mutation develop Phelan-McDermid syndrome. And by inserting this mutation into mice, actually Guoping had promising evidence that it could be affecting mice behavior in a way that had some resemblance to humans. But one of the biggest frustrations in this whole field is we base so much of our neuroscience on mice, but when we develop treatments in mice and we try them in people, they don't work, right? How many times have you seen this? Now you pick up the newspaper, oh, this disease has been cured in a mouse, that disease has been cured in a mouse. If you have Alzheimer's, you have autism, you're a mouse, you're in great shape. You go to a mouse doctor, hey, they can cure you right away, right? The problem is your person. Your person goes to the doctor and says, sorry, you're not a mouse, and because they're not working. And it's maybe not, so mice are so radically different from people, so maybe we shouldn't be so surprised that things that work in mice don't necessarily work in people. And so one of our, I'd say, a great hope here, because so far it hasn't led to a treatment yet, but we're on the cusp, is that by using primate models, primates have brains that are much more similar to humans. This is just showing the human brain compared to this is a large monkey brain and this is a mouse brain. And not only is it just the difference in brain size, but for a similar volume, we humans, and actually shared with other primates, have 7 times the number of neurons. Right? The circuits have 7 times the number of neurons. You can figure it's going to work a little differently than in a mouse, right? And so, and one of the parts of the brain that is most implicated in a lot of these psychiatric disorders is known as the prefrontal cortex. The prefrontal cortex area shown in green and blue here, which is huge in humans, is pretty big in primates and is minuscule in the mouse brain. And so we feel like we have a lot more of the circuitry to work with in a primate model. And so using the CRISPR method, Guoping has been able to insert these mutations into monkeys. And this first project is a collaboration with our colleagues in China. And this SHANK3 mutation has been inserted. It was inserted into the primate embryos, and then those monkeys grew up, and their behavior was tested. And we saw a lot of behavioral differences in those primates that resembled what's seen in Phelan-McDermid syndrome. Stereotypies. So, a big problem in autism, people develop these motor stereotypies. They may be different in different people, some with the hands, some with head banging. And it's the same thing in monkeys. Turns out they also develop stereotypies, but one monkey it'll be this thing, one monkey it'll be that thing. They're different, like people. It's not just one thing, but it's abnormal what they're doing. Sleep disorders, very common problem in autism, not just Phelan-McDermid syndrome, but many forms of autism. People complain that they can't sleep, and the same thing is found in the monkeys. That when you monitor, you put the activity monitors on them, either a watch, not the Apple Watch, but like a monkey version of the Apple Watch, or a collar that monitors their activity. You can see that their sleep is disturbed at night. They get up, and they have trouble going to sleep, and they wake up, again and again. That's found as well. And if you look at their social behavior, you can see differences as well. Now, this is a video of 2 normal monkeys. They're in separate cages before the test, and the technician comes in and he pulls out the divider So now the two monkeys, for the first time, get to experience each other. And, they're a little wary, they're not going up and hugging each other right away, but they're checking each other out, right? This is normal monkey behavior, is the monkeys try to get to know each other.
What gender are they?
Good question. I don't know in this video, but we've tried all the different gender comparisons, male, female, 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 and so on. Okay, so now we're going to do the same test, or show you a video of the same test, but this time there's the control monkey is over here, and the animal with the SHANK3 mutation is over there. And did I just mess up on that? Let me see here. I think I made it. Okay. Yeah, one of the things that, let me see here. One of the things I was going to show you. Oh, did you see that monkey flipping over and over again? Very abnormal. Normal monkeys do that very rarely. That's one of the stereotypies that this monkey developed, just flipping. So now the experimenter comes in, he pulls out the divider, and you could see this doesn't have that same kind of interest in the other monkey. He's nearby, but he's just looking off away. It seems not interested in that social exploration, which has some, again, reminiscent of some aspects of some of the social problems that we see in human autism. Okay. And then when we do MRI in these monkeys, we can look at both structural differences. It turns out they have reduced gray matter. But also we can look at the connectivity in the brain using MRI. And what we find is just a different pattern of connectivity in these monkeys. In some areas we see increased connectivity in local circuits, and in some areas we see reduced connectivity. But the areas where we see the increased connectivity tend to be sensory areas. And it's interesting, one of the things that people on the autism spectrum complain about a lot is increased sensory sensitivity. Very, very sensitive to lights and sound and so on. They really bother them. And what we're seeing is what could be a neural basis for this in the brains of these monkeys. And so, this is the kind of thing we need. We need biomarkers, we need areas to target, and we need things that we can go in, and if we test a treatment, we need to be able to go back in and look at the MRI and see how it's changing the connectivity, for example. This is all that monkey work was done as a collaboration in China, but we have here at MIT, we're not able to do work with those big monkeys in these genetic studies. There's just not even the space for it. But we've developed another primate as a marmoset. These are tiny little primates, really the size of rats. And so it's possible they live in big cages, many more of them. More fun than a barrel of monkeys here, I can tell you. We have our first marmoset colony here at MIT, and we're doing the first genetic manipulations, and so we'll be having a parallel program here at MIT to the one that we have in our collaboration in China. So I'm going to go back to the human brain imaging and talk about how we use brain imaging to try to understand what's going on in forms of brain disorders. I want to particularly focus on the work of Nancy Canwisher, Rebecca Sachs, and John Gabrielli, three of our faculty members here. Nancy Canwisher is the world leader in identifying the functions of different parts of the cortex. What she's shown is a functional localization for many different aspects of the processing of the sensory world. We have parts of the brain that are specialized for the perception of faces, shapes, language, seeing the bodies of other people, and so on. These are all these different colored regions here are the areas that Nancy has identified with these sort of functional specializations. And since one of the areas with these functional specializations are involved in face perception, so that as a possibility. You think that, sometimes people with autism, for example, complain or are seeing that they don't pay attention to faces or are afraid of looking at faces, don't look at the eyes of faces and so on. And Rebecca Sachs is taking this even further and looking at the functional localization of really advanced cognitive functions, including what she calls theory of mind. In other words, how do we understand what someone else is thinking, right? We do this all the time. We think, well, what are they thinking, right? And it turns out we have specialized circuits for that kind of thinking. And again, it was the kind of thing that you would hope that you might be able to get some insight into, say, a disorder like autism by understanding what's going on in these thinking about other people's minds areas. And I have to say that it's turning out to be very, very challenging so far, because a lot of these studies have not been targeted to the different genetic forms of autism. So a lot of this work we've been doing in just general autism populations, and there's so much variability in it that it's been very difficult to identify problems that are common across the whole, say, autistic spectrum population. But Nancy and her colleague Caroline Roberts have identified a perceptual problem in people on the autism spectrum that seems like it could turn into a biomarker. What she has is she has people looking at different patterns with the two different eyes. And normally, if you see two different patterns with your two eyes, you switch back and forth. You can't see them both at the same time. You switch back and forth between them. And the rate at which people on the autism spectrum switch back and forth is much reduced compared to the normal controls. And this is, people doing work in animals have identified this problem as associated with an imbalance in the excitatory and inhibitory transmitters in the cortex. And that's a common source of speculation about what's one of the problems in autism. And Nancy is now actively pursuing this as a biomarker, and Nancy and Caroline in autistic populations. John Gabrielli has been applying his brain imaging approach to understanding a variety of disorders, including dyslexia in children, and comparing dyslexic children with normally reading children. He's identified brain circuits that appear to be different between the two groups, involving many areas involved in language and perceptual processing and so on. And now, having identified these in kids who are already reading, John's now able to go back in and look at the connectivity of these areas in kids before they read. And so the goal is to try to identify kids who are going to later develop dyslexia and then target those for an intervention so that they get off in their reading to a much stronger start. And those are studies underway. And a related project we have, and I have to talk this one because it's one of my favorite projects that I've been involved with, and another collaboration with China. We've been trying to see if early music education, and particularly in this case piano education in kindergarten children, lead to generalized brain improvements in kids. And I don't know how many of you have children, or if you study music or your kids have studied music. You all know kids, study music as kids that are incredibly talented. Actually, when I came here, one of the things I was surprised at was hearing that MIT students, they may be engineering majors or science majors, but many of them have musical backgrounds and continue to take music instruction at MIT. Many of you are musicians, or at least as children were you musicians? Yeah. Very, very, very common here at MIT. And so the question is, was that a cause or an effect? Right? Were you attracted to music because you were a smart kid who was able to pay attention to music, or did early music education make you smarter and more focused? Right? It's actually a fundamental question. Well, yeah, it could be or they call a virtuous cycle. But you need a clinical trial, right? You need actually the only way you get an answer to these kinds of questions is you actually have to randomly assign people to the different groups. So in this collaboration with China, we were able to do that. We could randomly assign kids to either early piano education, additional reading instruction, a practical issue many schools have to face. Do they put their money into the arts, or do they put their money into more reading and math? So in this case, the comparison group was a reading group, and then another group of kids who got nothing, but at the end of the study, they were offered the free music lessons. So in the end, everybody got something. But we studied them before the control group got the music, okay? And we also took brain measurements using EEG to see how the brain responded to hearing different sounds and so on. So it turned out that the kids with the early piano instruction did the best, not only hearing the differences between tones, but hearing the differences between sounds and speech. And that's known to be impaired in kids with dyslexia, or at least one form of dyslexia, is that there's actually a speech sound processing problem. And kids actually are not hearing the distinction between words, and that's causing later reading problems. And so the kids with piano instruction were actually better at understanding words than the kids who had the extra reading instruction. It was really quite remarkable. And if you look at these brain signals, their brains were picking up these differences better than the other groups as well. So now the next phase of the experiment we're going to be seeing, is it the case, can we find the kids who the brain signals suggest are not picking up these sound differences as well, and would early music education give them an advantage to overcome some of these problems?
Question.
Yeah.
Is speech the feedback that's learned through hearing? And diagnose it using their speech?
So in some forms of dyslexia, you can give hearing tests that predict whether the kid's going to have a reading problem. That's right. Now, it's not, but one of the reasons it's controversial, again, just dyslexia is a heterogeneous condition as well. And there's only subgroups of kids that have that problem. They don't all have that problem. So, we're trying to understand, like, what's different about one subgroup compared to another. So I just want to give you we're coming near, we're approaching the end, about another example of how we can use social anxiety, use brain imaging to understand a disorder that would lead to better treatment. John's been looking at social anxiety disorder and looking at the differences in brain activation between people that respond well to treatment compared to people who don't. This is behavioral therapy for anxiety disorder. Some people respond really well, so some people don't. And it turns out you can identify these two groups from their responses to different perceptual images. And now John's trying to use that to make predictions about who will respond best to treatment. So in children, he's identified kids who are what we call at risk for depression compared to a normal control group. And the kids who are defined as at greater risk for depression are ones that have, for example, a parent who suffers from depression. So they're at higher risk. And he compares them to the kids with that don't have this high risk in their background. And he looks at their brain response while they're looking at images of other people's faces expressing different emotions. And it turns out that in the high-risk kids, they're showing a decreased response in some of these brain circuits when they look at happy faces, and an increased response when they're looking at fearful faces, as though they're actually more sensitive to the negative emotions in other people than the positive emotions. Now, these are kids who are showing, currently, they're not depressed. These are normal kids at higher risk, but they seem to be processing the sensory input differently than the control group. And so, then he uses just the brain imaging pattern to predict who will develop depression or anxiety disorders 3 or 4 years later. And he's comparing the predictions from the fMRI data, which is the yellow bars, compared to using behavioral symptoms for predicting whether they're going to suffer from these disorders. And it turns out they're more accurate and they're more sensitive, the brain imaging predictions, than even the behaviors that are related to what you're seeing in depression. So again, it's at least promising that we might be able to identify kids early who are at risk of developing depression and develop a better intervention. We're beginning to run out of time, so I'm going to skip ahead and say, so what's the roadmap look like over the next 5 years? What can you expect from these kinds of brain research studies? So I think it's based on everything that we've seen. Over the next 5 years, we're going to have a better genetic understanding of all these kinds of disorders I've talked about. We'll have much better understanding of the different subgroups that end up with the same label. We're going to have better animal models that are going to lead to it's going to lead to a better understanding that will lead to new treatments, and it will give us a way of testing those treatments in a way that we hope will be more predictive of how they'll work when we apply them in human beings. We'll identify the affected cells and circuits. We'll have new targets for drug development. I think we will see the first applications of gene therapy in brain disorders over the next 5 years. And I believe we're going to have better brain-based predictors of who's going to have problems either in education or suffering from different forms of depression, anxiety, and so on in kids, and that's going to lead to better interventions. So that's where I think we're going. These are again, this is the roadmap I think that's been successful in other areas of medicine. It's going to be successful in brain disorders, and I think we're just going to have to be patient for a few more years. Yeah?
Many of these disorders have low penetrance. Namely, if you have the genes, you don't necessarily get the conditions. Schizophrenia appears to be that way. Depression appears to be a way. Some are not, like ADHD. If you got the genes, you basically get it. But on the conditions with low penetrance, they have to be targeted in some way. Anyone studying what is the targeting mechanism from someone that is genetically vulnerable and either that gets it or doesn't get it, depending upon environmental or other factors, bad nutrition, war, etc.
So a good point. So you put your finger on a key problem, but I want to distinguish penetrance from heritability. So the penetrance issue is that for a lot of these disorders, we've identified many genes, any one of which has a very small impact. So you might have a mutation, but the probability you're going to end up with the disorder is very small. That's low penetrance. Okay. But the disease could be highly heritable because it's a lot of genes acting together. So the heritability of all these disorders is very high. So if you're looking at the transmission through parents to the kids, you can compare identical twins to fraternal twins. The heritability is up around 80% in most of these disorders. So we know there's a very strong genetic basis, but it may involve a lot of genes. And that is a key problem. So we have two ways of thinking about that. One is that when we go after the highly penetrant genes, which are being identified, that when we understand how they work, why does a mutation in this one gene lead to a high probability of a particular disorder, that we will identify the circuits that are being influenced by lots of other genes as well. And then once you identify that circuit, now you have a target for therapy, and it's going to help everybody, even with the polygenic forms of the disorder. So that's one. And the other possibility is we may need to model the impact of lots of these different genes. It's going to be much more challenging, but some of the new CRISPR-based methods allow us to target many genes at the same time. And so we will be targeting, the highest impact genes and see, how many do you need to affect? And is there a common circuitry that they're all affecting, right? So it's challenging, but I think, we have a reasonable path for how to study it in the future. Let's see. Well, let's go to the back. All the way in the back. Yeah.
Thank you. I have a question regarding the probability of children that had one clinically, let's say, depressed parent in terms of the environment. Because I'm very curious to see if the outcome of the increase in depression was actually due to genetics or also the environment created by having a depressed parent.
Yeah, great question. It's genes or environment. So, sometimes genes work through behavior. So in other words, you could have a genetic predisposition for creating an environment that leads to your children being depressed. In fact, there's a lot of support for that idea that one of the biggest impacts a parent has on their child is what environment they raise them in. In some ways, this is really difficult to tease those apart, but they're all working through the brain eventually, right? So if the kid's depressed, whether it's this, they got depressed strictly through a genetic cause or they got depressed because of the environmental influences, it's affecting, we believe, some of these circuits in a similar way. And so we're hoping that we can target depression using therapies that don't distinguish between whether you had a genetic cause or an environmental cause. But maybe in the end, we'll have to treat kids differently depending on the genetic basis. Let's see, lots of questions here. Let's just take the ones. We'll work our way from the back to the front for a change. So yeah. So the question was, why is it that the prevalence of some of these brain disorders is increasing over time? Because our genes aren't changing so much over time, right? So we have a 20% increase in childhood depression. Our genes can't be the cause of that. And so I did mention this at the beginning, is that we think we have better assessments of people now. So it's not the case that 20% more people are truly depressed than they used to be, but we're identifying people with depression better now. Likewise with autism, likewise with schizophrenia. All these disorders are being identified more. And as we reduce the stigma associated with different disorders, people are more willing to come forward, more willing to talk to their physicians about it and so on. And so now they're now coming up in the statistics. And what it's telling us is the problem was bigger than we thought, not necessarily that it's getting worse than we thought. Let's see, yeah.
Thank you very much. I just want to verbalize that Rotary International in 1987 initiated the eradication of polio. At that time, 250,000 people were getting polio every year. We're now down to 8 in the world. Pakistan and Afghanistan, and they hope to get rid of it in the next few years. If you're retiring and you're thinking of serving your community for the rest of your life, think about joining Rotary.
All right, there you go. Yeah.
I noticed you mentioned Alzheimer's. My family suffers studying it a lot. And one of the big theories now is that lectins, a poison that plants make to kill bugs, penetrates the intestine, goes up to the brain, causes the glial cells to strangle the neurons shrink, and you become any truth to that? Or you guys do? You must be doing some research on Alzheimer's.
Yeah, so you asked about Alzheimer's. Yeah, I was afraid somebody was going to ask about Alzheimer's. Yeah, and so you asked about a particular toxin produced by plants and whether that might be playing a role. Many of us were really, really, really hoping the amyloid hypothesis of Alzheimer's disease would turn out to be right. And we just had the worst news with the failed Biogen trial. And there are many people who are ready to throw in the towel on amyloid and say it's something else. And so, unfortunately, it's like we're back to the drawing board, and it's not really clear. So, environmental Alzheimer's actually has a fairly weak genetic component. So it's Some people call it, it's just, this is a form of aging, right? Some people say that, yes, it could be the toxins in the current environment that we didn't used to experience before and so on. It's really terrible that we're at this point so far into studying the disease that we haven't even figured out the cause yet. And so what you're saying, it could be a plant-based toxin. I don't think anything is off the table.
Your glial cells are fighting the inflammation.
So the glial cells are still very high up there in terms of people thinking about what's involved in the disorder. But what activates them or what causes them to work in an abnormal way, I think, still remains to be understood. Yeah.
Bob, would you describe Anne Greenville's study that was in Scientific American about creating an addiction which becomes ingrained,
Right.
and then she reversed it?
Yeah. So there's this idea that that addictions result from a hijacking of our normal circuits for we have. You could say, we become addicted to eating and drinking and things, right? We have our brains are wired up to strengthen behavior that leads to rewards, right, and diminish behavior that doesn't lead to rewards. And the problem is with some of the addictive substances, they act directly on those same circuits, and so it's like your circuit is now thinking that you're taking these drugs and that's leading to all these rewards, when actually it's the exact opposite, because you've hijacked them. And so I think it's that's a very active area of people trying to figure out how to unhijack them, or how to keep them from how to protect these circuits from being influenced in this way by drugs of abuse and so on. As you know, vaccines. People have talked about vaccines against addictive drugs. I think it's an area where I have to say where the progress in our understanding of the disorders are really gaining quickly. And so I'm becoming more optimistic that there are going to be real treatments that come out of it. Yeah, in the back.
Has inflammation got to do with Alzheimer's and this new thing about Pfizer squashing another trials? Is there any?
Yeah, I read that same news report, I think, that you did, that Pfizer had an anti-inflammatory drug that was used in another purpose, and they just, an incidental finding in the study was that it seemed as though fewer people had developed Alzheimer's than the people not taking the drug. I also read, so to be, to give them the benefit of the doubt, they had determined that the drug actually doesn't get into the brain. That's not a good sign. And there are other anti-inflammatories that don't seem to be protective against Alzheimer's. So I have to say, the problem with these drug companies is they've lost billions and billions of dollars pursuing Alzheimer's therapies. And they're becoming a bit gun-shy. In fact, that's one of the reasons we're so focused on developing the better primate models, is just to convince the drug companies to get back in the game, because they keep, they're realistically saying, look, we've been following the mouse work, it's not working, and so we're saying, so we're getting out of this field. They're laying off their neuroscientists. They're getting out of the business, and so what we want to do is we want to have. If we show that a treatment works in a primate, that's going to get their attention again. They say, well, now maybe it's worth reinvesting the money that's going to take to do another trial. But that's where we need to get. We absolutely depend on the drug companies. We got to get them back in the game. Yeah. So I could not. I'm having a hard time hearing you too. Oh, periodontal disease. Yeah, I know. I read that too. I was really excited about it. The possibility. I'm flossing more. I'm definitely flossing more. Yeah, so I know. Very, very, very interesting research. Showing that there's a relationship between periodontal disease and Alzheimer's, and they developed a small molecule that target that bacteria better than the broad spectrum antibiotics, and they seem to have a positive impact. I know that those are going into clinical trials from some of those companies right now.
The kind of study that being done, the data that's being shown, the correlation that's being countless, and I'm just surprised that it's not even discussed among the public, like the patient population.
Well, so the thing is, I think it's another example where I think I, along with a lot of other people, got really excited. But the problem is our hopes have been dashed so many times that.
That's preventative, that's a risk factor.
Right.
I'm just saying.
But anyhow, but the trials are going to happen, and we will see. Absolutely. Yeah. Let's see. How about there in the back?
Has there been a lot of research on because we live in a day and age where there's increased toxic burden in our blood systems and stuff. Has there been research on whether or not the blood-brain barrier has been under more assault in the last few decades?
So the question was that we're subjected to so many toxins, many more toxins these days than people were in the past. And is it possible that our blood-brain barrier, which is the way our brain protects us from all these toxins in the blood, that it's under more assault these days? That's a very active area that people are working in. There's some Parkinson's disease is an example where the most prevalent ideas. There's a lot of Parkinson's is due to toxins. Which toxins is another story. That isn't quite figured out. But Parkinson's is an example. When I said where a lot of this, the increase in incidence of disease is due to better assessment. Parkinson's disease seems to be a disease that just didn't exist in the past. It wasn't even an assessment problem. It actually seems to be a modern disease, and that lends itself to these sort of toxin hypotheses. The modern humans are being exposed to toxins that people in the past were never exposed to before. And so I agree, that definitely needs to be is an active area of research. Carol,
Maybe time for one or two more questions?
Right here.
I saw that you had a slide on mindfulness. Is there any mindfulness research for treatment of depression that's being done at MIT or?
So your question allows me to mention that. I skipped that over because I was worried we were running out of time. But yeah, John Gabrielli has been using mindfulness as an intervention in kids that have been identified as at risk for developing depression and anxiety. And it seems to be helpful.
And what's it doing in the brain to be able to prevent depression?
So I don't know the details of this, but I think that what John's doing in terms of looking at the brain's response when you look at faces of people carrying negative emotions and positive emotions, he's seeing how they change with the mindfulness training. So it could be that as kids learn to better control their own emotion and their own levels of anxiety, it's changing the brain's response to these, positive and negative emotions from people on the outside. So I don't know that specifically, but that's where I think John's going with that.
Is that better than microdosing?
Microdosing of what?
Yeah.
Huh?
LSD.
Oh, LSD?
California and microdosing,
I don't know that. Right, one more question. Yeah.
Going back to the Alzheimer's question, are you familiar with the work of Dale Bredesen and the concept of Alzheimer's maybe, or dementia as being really not unifactorial but multifactorial, and the idea of trying to target them with one treatment not making sense because you have multiple things coming in? I don't know.
Yeah. So and the idea that there's multiple factors that are all adding up to to giving people Alzheimer's is certainly a viable idea. So when you look at people with Alzheimer's, there's a lot of other things wrong with them. They may have a buildup of amyloid in the brain, but according to research, 99% of people diagnosed with Alzheimer's have vascular disease in the brain, and so there's going to be a lot of factors weighing on the development of vascular disease. So it could be. I think we need to.
We're going to wrap up. Let's thank Rob Desimone.